For us, I believe we stopped number 5. Should we call cytoplasm 5? Okay. Cytoplasm is like just in eukaryotes, right? We usually list that it's mostly water, 80% water, ions, right? Dissolved salts, small organic molecules, right? So the building blocks, the monomers. Just to kind of, make you practice chapter 2, meaning amino acids, monosaccharides, lipids, right? The nucleotides of nucleic acids, the building blocks of nucleic acids. And in bacteria, what takes up most of the cytoplasm is actually the DNA, is actually the chromosome. So I think on the handout, I have them as separate numbers, but I think I'm just going to keep them under the cytoplasm and letter them this time. I hope that doesn't throw off your whole organizational scheme. But the chromosome does take up most of the space. And again, I would remind you, over and over until this is tattooed in your brain, right? Bacteria, prokaryotes, have one single chromosome. And it is circular, right? It is still made out of DNA. So it's still made of nucleotides, A's pairing with T's, C's pairing with G's. So it's still complementary. It's still double-stranded. It's still a double helix. Right? What it does not have that eukaryotic DNA has, it's not associated with very many histone proteins, and it's the histones that wind the DNA up and make it so that it fits into those fuzzy chromosomes that you see in eukaryotic cells. So in a bacterial cell, I'm always drawing it like this. Later in the semester, I'll draw a bacteria like this, and I'll do this to represent the chromosome, right? And I'll try to draw little lines in it, and I'll draw it loopy to remind you that it's one chromosome, it's circular, it's double-stranded, it's a double helix. But in real life, this chromosome takes up most of the space. So in real life, it wouldn't look like this. It would look more like this. Right? And it would occupy most of that space. It's expanded and available for transcription, almost the whole chromosome. Okay, nonetheless. Obviously, the function of the chromosome would be that has the genetic code on it to make all the proteins. And of course, this is how the bacteria reproduces or passes on. It's hereditary information, if you want to think of it that way. Bacteria also often have, sometimes have what we call a plasmid. And a plasmid is another piece of circular DNA that's a lot smaller. Just to give you a little reference, usually say E. coli has about 4,000 genes, right? You have about 25,000 genes, right? So that's how they can all fit on this much smaller organism. But a plasmid just has a handful of genes, 10 or less, maybe even. And we refer to it as extra chromosomal DNA, right? Not part of the chromosome that's supposed to be E. But it is also circular, usually. And it's also double-stranded, double helix. I'm not going to bother to write all that. I like to remind students what is on the plasmid are bonus genes. It's never something that I shouldn't say never. It's usually not something the bacteria absolutely has to have in order to survive. It's more like not just having a handbag, but having an expensive handbag, or not just having a car, but having a really nice car, right? So plasmids have things on them like, ooh, resistance to an antibiotic. That would be good if I was the bacteria, right? Good for me, bad for the host. Ability to make a toxin that might help the bacteria, multiply more in the host. Maybe it's the ability to make fimbriae, right? Maybe it's some other feature that bacteria normally wouldn't have, but if it has it, it's going to help it survive a lot better, a lot faster, reproduce a lot faster, make more disease. Always included in the plasmid are the genes to make apillis. I'll put pili. Remember we talked about pili and fimbriae, right? Natopilin. And the pili in particular are to extend to a neighboring cell so that you can give them a copy of all of your bonus genes. How cool is that, right? It'd be like if I gave Alex a copy of the test before class and she had no, and I won't, don't need to impinge on your integrity, but let's say she had no scruples and she just copied it and gave it to all of you, right? It's just like that. Bacteria live in a big pile. And so as soon as one bacteria has the plasmid and has the ability to give it to their friends, then it comes with the ability to give it to their friends and so on. And soon all the bacteria in the population are resistant to whatever drug you're taking, right? And that takes about 2 days, right? Because they multiply every 20 minutes. So it's really quick. Okay. What else is in the cytoplasm? Ribosomes. I would remind, ribosomes have two, they're not membrane-bound organelles, which is why they're on this list. They have two subunits, large subunit, small subunit. And what's the function of the ribosome? No. Proteins, right? It's the place for protein synthesis, right? The location, the site for protein synthesis. It is important for you to have just a little bit of a knowledge that these ribosomes are a different size or a different density than your ribosomes. When the ribosome is assembled in bacteria, we call it a 70S ribosome, and the two subunits are a 50S and a 30S. Yes, I know that doesn't add up to 70. Your ribosomes are larger and more dense. And the reason this is important at all is because there are bacteria drugs, antibiotics that will target bacterial ribosomes based on their size and their shape that do not disrupt your ribosomes. So that's an advantage because you're going to be damaging the ability of the bacteria to make protein, but not the ability of the host to make proteins, right? And sometimes, so ribosomes aren't always, all bacteria have them, right? Plasmids are sometimes. This is also a sometimes. Some bacteria have what we call inclusions. These are areas of concentration of something, and it's different depending on the bacteria. They're usually elements or molecules, let's say, needed for metabolism. Sulfur granules, for example, if it's a sulfur bacteria, might be, let's just leave it at that. They usually appear in bacteria that are in the environment because they can't always access some of the things they might need for their metabolism, right? Because they're in soil or, laying on a rock instead of in water or in a body, right? In an organism. They're useful to us as microbiologists because most of them stain pretty colors. So it helps with identification of particular bacteria, right? So if you stain with this one dye and you get big pink granules, then you know, oh, that's a bacteria that has sulfur inclusions. It must be a sulfur bacteria, right? There are these cool bacteria that have, there's a picture of them in your book. I don't have it on the PowerPoint, so there's a picture. There are some bacteria that have iron oxide as their inclusions. And what that means is they have a row of like mini magnets inside of them. So the bacteria are always pointing north, right? Kind of cool. So I think they're photosynthetic. I think that's why they point that way, so that they can get light to make ATP, but I'm not positive. So that's kind of cool. Occasionally, there are very few bacteria. Some of them have what we would call sort of like actin filaments. So they sort of have a little bit of what might be considered a cytoskeleton, but that's kind of, you know, few and far between. I think that's it for the cytoplasm. Our last structure of prokaryotic anatomy is something we call an endospore. This is also, some bacteria have this, most do not, or have the ability to make this. So, and again, most of these are also environmental bacteria. So bacteria that are living in the soil, like Clostridium, like Bacillus. Those are two big spore formers. The bacteria normally, right, living its bacterial life. So I'm going to draw that DNA in there. And, you know, like I said, just living its life. When the bacteria, if they have the ability to make the spore, an endospore, those are genes that are on their chromosome. Okay, and it's a stress response. I have a test. No, it's a bacteria, right? So it's water usually, and temperature are the big stressors. Right, so I'm going to put decrease in water and the temperature, depending on if the bacteria likes to live in the cold or likes to live in the hot. That could be either way, right? And quite literally, when those stressors happen, they turn on a set of genes that are called heat shock proteins, because that was what they were first identified as a response to. But basically what they do, that doesn't show very well, so I'm going to get a different color. They send the bacteria into this sort of stepwise process to sporulate. So the bacteria make a copy of their chromosome as if they were going to divide, but rather than undergoing binary fission, they lay down this super thick spore coat. And it's not important right now for you to know what compounds are in that spore coat, right? And then the vegetative cell, or the actively growing cell, dies. So if you see this under the microscope, you literally will see it. You'll see all the bacteria, right? All your Bacillus subtilis will have these things in them. And sometimes they're located in the middle, and sometimes they're located on one of the ends, right? They're central or terminal, or we say subterminal. So when it's inside the cell, that's when we call it an endospore, right? When the vegetative cell dies, the actively growing, you know, living its bacterial life cell dies, all you have left is what we call I always thought this was funny, because a free spore, like Free Willy, free spore. And these free spores, they are very resistant to harsh conditions, such as they survive boiling, they survive freezing. Think about it, because they're in the environment. They also can survive most disinfectants. So to eliminate the spores, excuse me, if a culture has spores or is making spores, to eliminate those spores, you need to do like drastic things. Like you need to raise the temperature to 121 degrees Celsius under pressure, 15 pounds pressure to eliminate the spores, or you need to irradiate them, or you need, but mild irradiation is no good. They're resistant to UV light, which a lot of places use, public places use as an antibacterial, right? So like in a, sorry, my brain is not working very fast today. No coffee, so neurons firing a little slower. So like a lot of food service prep places have UV lights that are on or that they turn on when they're not in use, because that will inhibit most bacteria. Hospital emergent operating rooms in hospitals, when you leave, a UV light comes on because that will limit the number of bacteria growing on the surface, even though everything in there is metal and bacteria don't grow very much on metal, right? But spores, they're resistant to UV light, so sunlight as well. That's, I mean, it's cool for the bacteria, right? So like you can find them like on mummies and stuff, and like this ice guy they found in the Swiss Alps. It was before you were born, but they found this guy, like the skeletal remains of this guy in the Swiss Alps, because there was some big melt, and it was like he was walking, you know, from one little village to another, and so they had like, okay, I won't really call him shoes, but he was like, I don't know, it was like the skeleton was like 3,000 years old or 4,000 years old, right? So he was like mummy age, young mummy age, but in a different part of the world where we didn't think people were living yet, right? Where humans weren't walking around yet. So they took, there was like straw stuffed down in like pieces of leather or cloth wrapped around his little bony ankles, right? When they took the straw, they found spores on them, right? Like you can find bacterial spores in amber. Cool. And then you can see, right, what the bacteria were like 4,000 years ago compared to the bacteria today. Okay, they're not, that's not exciting for you, but I think that's really cool. Like if you're archaeologist, right, and you find like the food pit where the privy where they did all their business, right, you could get bacteria from there and they could tell you things about changes in the bacterial genome. Okay, not all bacteria do this. I would remind you, it's not reproductive. We have one vegetative cell. It makes one spore, and then the spore remains dormant. You can think of it until conditions get better, until those stressors are gone. And we go back to one vegetative cell. And it's quite honestly, guys, it's almost always water, which is why you find them a lot on mummies, because that is a very dry part of the world. And what happens is literally water penetrates the spore coat and the cell like expands and goes back to being one vegetative cell. Now remember those things you used to beg your parents to get, like when you were going out, when you're like after the grocery store and you were leaving ShopRite, and there were all those little vending machines for gumballs and stuff, and there were those little things that were like in plastic, they looked like a pill, and you would put them in water and they would expand like into a giraffe or a dinosaur or something, like a little sponge. Remember those? Yeah, that's what this is like, right? So it's like the little thing that you can't tell what it is, and then when the water starts to come in, it plumps up and becomes a bacterial cell again. And again, this is a sometimes, not all bacteria can do this. Let's see if I can find a few pictures to show you. Yeah. What happened to those? Okay. weakness. There we go. Yeah. So this I always feel like I'm one of those engineers in NASA, like in the 1960s, trying to use these buttons. Like this is like my Dr. Spock control panel over here. All right, so just like, really? That's what we have? Okay. So this is a picture of bacteria and the DNA is stained yellow. So the point of it is to show you how much space the chromosome takes up. And I know in most of these pictures, it looks like it's in two parts. That's just because you can't see all the strings in between the concentrated parts. Okay. So like this one, if you look at this one down here, you see how it's like taking up almost all the space inside the cell. Ribosome. There's the sulfur granules. I don't have the cool magnetic ones. I have to get those. And then this is supposed to be a picture of an endospore. They sometimes do look like safety pins, but you have to notice it's fluorescent microscopy there, right? So most of the time they just look like I drew it, like you'll see a circle within a circle. And you'll start to see them anytime we use bacillus now, because we got them out of the refrigerator, we rehydrated them in January, and now we're just making subcultures of them, right? And so like I made a subculture that we used for our gram stain. I made that on Monday. Now it's Thursday. That's really old to a bacillus, right? Because remember, the old is 24 hours old. Right. So now if we did the gram stain today instead of Thursday, that's why I was so anxious to do it on Tuesday, because I was like, well, they mostly won't see spores on Tuesday, but by Thursday, when you stain the bacillus, and it happened to some of you already, right? You get this like kind of double effect inside the cell, sometimes like that. You see purple and red with inside the cell. What you're seeing inside the cell, that's a spore already. That's an endospore, even though we're not staining specifically for the spores. So anytime you see that, hint, hint for your unknowns later, anytime you see that, that's a good indication it's bacillus and it's a spore former. You know. This picture is, I like better. It's different than the one that's in your textbook. This is about the morphology derangement again. And we did talk about that last time. Yes. Right. I put it in, I think toward the beginning, but the older version of the book puts it in at the end of the chapter. The newer version puts it in at the beginning. I like this picture a little bit better because it does show you some of the possibilities, right? So even though we think of coccus as being perfect spheres, these are some of the different arrangements that they might show up in: diplo, staphylo, strepto, and then sometimes they're actually almost like in squares. And it shows you also kind of the range of the rod or the bacillus shape. E. coli, which we looked at on Tuesday, that is a cocobacillus, technically. It looked very rod shaped for us because we've picked an E. coli that looks more rodish. But again, as I keep subculturing it through the semester, by the time, if you get E. coli for your unknown, it might not look so rod shaped. It might look more like a roundish rod, like a cocobacillus. Okay. And then the different spirals, because we won't see those under the microscope. And those are just some cool scanning electron micrographs, because I like those. I don't know what that is. Oh, that's oscillatoria, to remind us that cyanobacteria are also bacteria, but live in filaments. I think that's the cytoskeleton, like the actin. This particular bacteria has some actin in it and it's stained green. Oh, and those are some cool sulfur bacteria. I just like those. I don't know what that's about. Okay. Now, the last tiny little bit of this chapter is about archaea, or as your book calls it, the archaea. So we're going to mention them, and then that'll be the end of
chapter 4. The other part that's at the very end is about classification of bacteria, which I talked about earlier also. So we're almost finished with this one. And the archaea. Right. Which I usually spell wrong, but I did not today. All right, so we think of archaea as the extreme bacteria, or literally your book, it refers to them as extremophiles, loving extreme conditions, right? And they do kind of fall into 3 main groups. I don't know the names of the kingdoms. I don't think it's important for you to know them. They do kind of fall into 3 main groups, and the one of the groups is called the methanogens. Like that's their, their street name, their slang name, if you want to think of it that way. And these bacteria do something very unique in that they take carbon dioxide and hydrogen gas and convert it into methane. What? So they tend to live in places they're usually where there is not a lot of oxygen, right? And this would be the reason this whole group is called the archaea, is because these extreme conditions I'm describing, originally scientists thought, well, these are the same conditions we know exist or we think exist on early Earth. So we think these might be the closest living relatives to the first cells on Earth. So we're going to call them ancient, right? We're going to call them the archaea. Yeah, not true. Many years later, many decades later, what scientists found out is the archaea do things more like the eukaryotes than the bacteria, but they're still weird, right? So they're still interesting to study. They still do survive in the conditions of early Earth. They do give us like clues as to how the first cells might have made their way, but they aren't the closest related thing to the first cells on Earth anymore. But we're not going to rename them now. Okay, so we have the methanogens, which, like, thinking about those Stanley Miller-Urey experiments, right, with the boiling and having carbon dioxide and hydrogen and lightning, right, and making cells and amino acids. And the other cool thing is people are trying to figure out how to use methanogens to digest. You can use methanogens to produce more methane, and then the methane can be burned for fuel. It's not great for our carbon footprint, but it does kind of recycle basically a bunch of cattle feces, right? A bunch of dung. The extreme halophiles. So they live in a super high concentration of salt, like 30% plus. Again, so like conditions would have been on early Earth, on rocks and various formations. So now we would find them maybe in a cave or a salt mine, or we would find them maybe sometimes around where there are hot springs. And the third group are the extreme, I think we still call them extreme thermophiles. They live at temperatures above 80 degrees Celsius. What? Remember, water boils at 100 degrees Celsius. So where it's really, really hot. Hot springs, volcanic vents, above the earth or under the ocean. That's where you find the thermophiles, extreme thermophiles. So they're worth a mention there. And like I said, then there's some things about the taxonomic scheme of classification versus the clinical scheme of classification, which I know we already talked about, so I'm not going to go through that again. I would point out, because I don't know if I was recording that day. Did we talk about these questions at the end of the chapters and the green boxes and the Bloom's taxonomy and how to study for the test? Yeah, okay. All right, I won't go over that again. And let us then turn to chapter 5. Okay, so chapter 5 is a survey of eukaryotes or eukaryotic cells. Right? And the whole first section, that's review on your own. I am not going to go through, like, make yourself a list of all the organelles and their functions. Structure and function, right? That's bio 1 stuff. And again, you're supposed to get it in anatomy and physiology, although I know a lot of them skip it, but whatever. Take 10, 20 minutes to go through that. I'm going to ask you multiple choice questions only about the structures and functions of eukaryotic organelles. And I will do it in like 2 questions, right? Which of the following eukaryotic organelles is mismatched with its function? And I can give you 5, and only one of them is incorrect, right? So I can do chloroplast, mitochondria, endoplasmic reticulum, Golgi, and a vesicle, or peroxisomes, right, in one fell swoop in one question. So don't spend a ton of time on it because it's going to be multiple choice questions, maybe 2, maybe 3 to stretch, but have it at the ready. I mean, I would bother to go through and at least review that yourself. I don't think I have any questions about mitosis and meiosis, and I know that your book goes through those again. I would say it's probably, and oh, look, your book does that at the end of the section for you anyway. I would say it's also important to focus on the organelles and / functions that bacteria and eukaryotes share, right? Like some of them are photosynthetic bacteria. So remember things about the chloroplast, right? The flagella are different. We talked about that already. So that might be something that I would, since I bothered to spend the time to say those things, that I would ask you about. Okay. The second part and the third part of chapter 5, I guess it's section 5.4 starts with, is a brief review of eukaryotic organisms that cause disease in humans, plants, and animals. So fungi, protozoans, and some animals at the end. Okay, so we're going to just do a little overview here. It's not that I expect you to remember the names of these diseases and the names of the organisms. We'll meet them again if they cause disease later in the semester. But fungi, they often get overlooked. So we're going to have to give the fungi a little time. So some general characteristics. Right? Obviously, they're eukaryotic. Not going to write that because it's in this chapter. They are chemoheterotrophs for their nutrition. So dig deep back in those memory banks, right? So a heterotroph on the surface is an organism that can't make its own food, right? But in particular uses organic molecules to produce its energy. And the chemo part means it uses organic molecules or chemicals to produce its carbon, right? So it's organic molecules. Then it may break those organic molecules down to make ATP. So they use chemicals, either organic or inorganic, to both as their carbon source and to make their energy. And those are kind of 2 separate things, right? Making a protein is very different than breaking down a protein to make ATP. In particular, they belong to a subcategory of chemoheterotrophs that we refer to as saprobes. It's just one of those biology words you have to know. And no fungus, right, when you see it. Saprobe specifically means they feed on dead organic matter. Which, again, I don't know about you, but I am a meat eater, but I'm not eating like a live chicken, right? But I think they mean like, I think, the general sense is decaying things. Which brings us to one of the most important uses or purposes of fungus on the planet is that with the bacteria, with prokaryotes, they are responsible for most of the decomposition of organic material. And again, this is going to be important because we need to get all those nutrients back into the soil or back into the water so they can be recycled. That's enough about their nutrition. They come both in microscopic and macroscopic versions. Sometimes a single fungus will have a microscopic part of their life cycle even, and a macroscopic part of their life cycle, right? So you've all seen the mushroom, that would be a macroscopic example. Most of you have not seen streptomyces, which makes streptomycin, which is an antibiotic, right? We haven't seen that because it's microscopic. They are categorized or put into groups based on their reproductive structures. I think this is changing, at least that's what I saw in this edition of Campbell, but for right now and what's in this book, we'll stick with it. It's particularly the reproductive structures for when they do sexual reproduction. Now, this is the other funny thing about fungus, is that sometimes the spores that they make that are N, that are a result of meiosis, under the microscope, they look just like the spores that are N that they made to make more of their like sporophyte form or their big, macroscopic form. So sometimes it's very difficult to classify a fungus or even identify a fungus because you have to have it go through its life cycle so that you can look at the spores and see what the ones as a result of sexual reproduction look like versus the ones that are just asexual reproduction look like. Okay. So go to mycology, learn all about that. Right. We call sort of the multicellular part of a fungus. Multicellular. Right, so we call those hyphae. I think the plural or the singular is hypha. And that's basically, right, like a thing like this, like a row of cells, right? A filament. Sometimes, see, I've drawn this line between the cells, right? Sometimes the hyphae are separated like that into cells, and we call those septate hyphae. And sometimes they're not. So sometimes they kind of look like this, like they almost have a cell division, like a cell wall in between them, but they actually don't. And when this is obviously called non-septate hyphae. Why am I telling you this? Because you can use it to help identify what fungus you're looking at. And in the case of non-septate hyphae, sometimes it looks like some cells have multiple nuclei because the nuclei can get out and move in from one cell to another. There is also a thing called pseudo hyphae. Fake hyphae, right? That happens usually with a fungus that buds. I'm thinking particularly yeast. So you have a giant cell, and then you have like this smaller cell and the smaller cell and the smaller cell off of it. And it looks like it's a filament, but when you look at it with a powerful enough microscope, you can see they're actual individual cells, right? So like I'm compelled to mention that. Blah, blah, blah. Why do we care about fungus? Why do we have to have this discussion at all? Okay, number one, some fungus cause disease. Think big, right? Bigger than athlete's foot, bigger than mold on your strawberries. I mean raspberries, because they mold like the next day, right? Think like devastating crops, trees, right? Blights, smuts and rusts. I just like to say that because it sounds funny, right? And obviously diseases like ringworm and fungal diseases that are respiratory now because you can inhale them and then the spores. So I'll put humans and animals. Some fungus produce deadly toxins. Some of the most deadly toxins on the planet are not man-made. They come from fungus, and then they are modified from a fungus. Insecticides, for example, there's a whole bunch of them, but you've all heard tales, right, of someone eating a mushroom that was actually a toxic mushroom and not a mushroom that you should eat, and then it degrades your liver and you die within a week unless you get a liver transplant, right? So like seriously powerful toxins, right? Not just ones that can give you hallucinations, but ones that can actually kill you, right? So be wary. I'm going to reiterate the decomposers because we're going to, fungus are also, not all fungus are bad. Fungus are our friends, right? We use them for food, right? And to produce some of our food. Alcohol is all made by fungus, basically, right? It's yeast fermenting something, eating it, and then producing as a byproduct alcohol. Food, alcohol, cheese, yogurt, right? Bread. Ooh, bread, yum. Bread. My list. I don't think there's any others that I have to put on there. Oh yeah, I should put that too.
Sure.
Number 5. Not going to spell this correctly. I already know it. Micro. I think it's like that. Actually, it's no R. I think it's micro. Micro. Not sure. You should look that up. There's 2 H's or 2 R's. I don't really remember. These are the fungus that associate with the roots of pretty much almost all plants. And they're totally symbiotic. They live inside the root hairs, and they are totally helping the plant get minerals from the soil and water, believe it or not. So, for example, some major efforts at reforestation, for example, right? So like, oh, the logging industry came in and wiped out this forest, so we're going to replant it. Great. They take a bunch of seedlings and stick them in the ground, but if the right fungus isn't in the ground, they're all going to die. Right? It happened many times before they realized you need more than just the plant. You need the whole plant environment, the plant's whole environment. And depending on how disturbed it's been, right, the fungus in theory might still be in there, right? If it was just logging, but if it was like mining, where we cleared the trees and then dug up the earth, they're not going to be there. So that's important. Number 5, that's good. Okay, so that's enough about fungus. Let's talk about the protists a little bit. They're some of my faves. Pretty good. Try to remember they too are their own kingdom. We tend to divide them up into the algae, the green ones, right? And the protozoa, those are the ones that are more animal-like protists, right? There are a few of these that are that cause disease. I'm thinking like trypanosomes and Giardia. There's one amoeba. Most of them are free living. The important part I need to tell you about the protozoa, especially the ones that cause disease, is similar to a endospore. They have a life cycle where if conditions are harsh, they have an actively growing form that's called a trophozoite. That's like their feeding form. And when they encounter harsh conditions, many protists can form what's known as a cyst, C-Y-S-T, and that is similar to an endospore in that it's a resting structure, right? It's super resistant. Some of them you find are resistant to chlorination process in water purification, or the UV light treatment in water purification, or you have to use both in order to get rid of the cysts, or it's still in the water. So all those like nature shows and survival shows where you see the people like dipping their hands into the clear stream and then drinking it, don't ever do that, right? Because there are protists in there and cysts of protists in there. And when you do that, they get into your digestive system and they cause some really nasty diseases, right? Particularly Giardia. Lasts for about 3 weeks, even with medication, right? But these cysts are usually not as resistant as endospores. Like if you boil the water, it will get rid of the cysts. Or if you filter the water, the cysts will get trapped in the filter, right? Or treat it with iodine or some other halogen, you can get rid of them in the water. Algae we put on our list to remind us, right? So these guys are all photosynthetic. So they are big oxygen producers for the planet. Some of them are also nitrogen fixers, although that's more of those cyanobacteria. They are, let's say, health monitors or indicators in bodies of fresh water. Yeah. Bodies of fresh water. Meaning lakes, streams, etc. Right? If there's a bloom of a particular algae, that usually means there's too much organic material in that water, either because it's being dumped there by a chemical plant, or it's runoff from a farm, or it's animal feces that's untreated, and that's causing a bloom or an overgrowth of a particular kind of algae, which will have a negative impact on that water ecosystem. Okay, so they're really important for that, right? With what algae is growing and how much of it is growing at what time of year. Oh, and some of these make awful toxins too. That's a good story. I don't have time to tell you, but you can probably find the book on Amazon. It's called, I think it's called "And the Seas Turn to Red," and it's about this bloom of this algae that attacks fish. And if you eat the fish, you can have neurological damage and be paralyzed. It's a good story. And it's all because of basically pig farming and the feces of the animals running into the river, into the water. All right, so protozoa, we talked about a few. Yeah, they can cause sickness. Ooh, and then let's talk about the helminths real briefly, and then we'll call it a day. Okay, helminths, that's again, it's a slang word. It's an abbreviation for two phyla of animals. So these are actually animals, just like you. helminthes. I think that's where the roundworms are, like the nematodes. Wow. Ascaris, whipworm, pinworm. And ash helminthes, I think it's spelled I always spell that wrong. I have to look. ASCH. Yes. These would be your tapeworms and your flatworms and your flukes. Okay. So you've all seen, this, an amazing tapeworm, right? Okay. So just briefly, as a reminder, they're animals, they're parasitic animals. Most of the time, humans, I'm going to say this, are rarely the definitive host. And what that means is the primary host of the organism, the host that the organism reproduces in best, right? Most of the time, humans are an accidental host. We eat something that's not cooked, basically, right? Or we swim someplace where there are things that can burrow through the soles of our feet. That's not good. Mantra of microbiology: wash your hands, cook your food, seek prompt medical attention. No swimming with snails, right? Because those are, snails are the definitive host of the ones that burrow through the soles of your feet, and then you cough them up, and then they go into your lungs, you cough them up and swallow them. It's gross. So wash your hands, cook your food, seek prompt medical attention. No swimming in bodies of fresh waters with snails. Okay, remember that. If you learn nothing else in this class, that's what you should learn. But of course, you can get them from all different kinds of things. So we're going to just mention briefly, again, the tapeworm, right? Those are the ones that pretty much most segments past the head or the scolex are reproductive segments. So basically the tapeworm can just constantly produce fertilized eggs, and then as the segments break off and are released in the feces of the animal, other animals eat them, and then they get a tapeworm, right? No, you can't coax them out by holding a bowl of milk in front of your mouth, right? That doesn't happen. They have to come out the other way. There are drugs that you can take, right? The challenge is they are endemic. You got to have clean water, and water that's not contaminated with human or animal feces, right? So either water treatment or wells and sanitation enough that basically you aren't defecating where you're drinking your water, right? So public service announcement: buy the toilet paper. Who gives a crap? Because they give 50% of their proceeds to building toilets and privies in countries where people don't have clean water. Plus it comes to your house in a box and has cute little wrapping paper on it, and it's fun. All right, tapeworms, flatworms. Oh, the roundworms. Let's just look at the pinworm for a minute because it's so cute. Right. Maybe I have that. It's in your textbook. Oh, they went from line drawings to actual little children. Oh, how cute. So up here on the top of it's figure 5.25, and you see this little girl here, and she has a pinworm in her digestive tract. Pinworms are a nematode, so they're in that first category. They're roundworms. The male actually lives inside a groove in the female worm, and so as the female worm produces eggs, they're fertilized like constantly. The fertilized eggs travel down the digestive tract and end up in the feces, right? And the little baby worms, they can either be excreted, or they start to set up shop around the anus, which gives little children scratchy bottoms, right? So when your parents or grandparents would say, "Do you have ants in your pants?" That's basically what that means, right? they're diagnosed by you take a piece of tape, literally, and you put it across their little bum holes, and then you lay that on a slide, and you can actually see the little worms and the fertilized eggs, right? You can be treated, it's no big deal. Once you have children, most of you are kind of on the young side, but once you have kids and they go to kindergarten or first grade, you get this letter home, you know, someone in your son/daughter's class has whipworm or pinworm. Please check your child for an itchy red anal opening, right? And if they have one, seek attention of the doctor and let the nurse know, because it spreads like wildfire, because little children, right, in daycare and in kindergarten and first grade, they scratch their bottoms, don't wash their hands after they go to the bathroom, and then stick their fingers in the mouth, or they put them all over their pencil and then generously give that to their friend to use as their pencil, right? When their friend loses their pencil. Or you see these two little girls, you know, giving each other a hug, sitting in a sandbox, right? Okay. You know, or on the pavement, right? So it spreads very easily back to themselves and to all their little friends, right? So, yeah, it's just to kind of give you an idea of the life cycle of a worm. And that's the end of chapter 5 for us, as far as I'm concerned.